I've spent the last couple of days updating the PowerPoint, turning the background to white so it can be printed. Some of you are helping me with that. Putting in new slides so there'll be more stuff to ask you on exam 4, stuff like that. So I'm going to kind of backtrack and cover the new stuff I inserted because I think it's very important. You know, as we went from the discussion, I think this is on, but maybe it's not. You know, it was not. As we went from the discussion of how DNA is packaged in bacteria into eukaryotes, we forgot a definition, and that is the definition of chromatin. So I took it here out of the dictionary. A complex chiefly of DNA and histones in eukaryotic cells, usually dispersed in the interphase nucleus. It's condensed into chromosomes in mitosis and meiosis, and in which the nucleosome makes up the fundamental repeating unit, chromatin. I had a question on my written PhD prelim that I still remember because the guys didn't give me credit. And of course, I was in transcription and chromatin and all that, just lived and breathed. And they said, what are the components of chromatin? I put DNA, histones, but I didn't put RNA polymerase. It used to be that the definition of chromatin was more operational. What did you find there when you spun it down and purified it? What you found, everything. You found a little RNA, and now it's gotten more towards structure and organization because it's kind of trivial that all that other stuff is there to regulate and various unknown functions. Anyway, I missed it because I didn't put on RNA polymerase. Of course, that's what the lab I was in, that's what we studied all day. Well, of course I shouted to the heavens. Anyway, it didn't scar me, and it hasn't made me bitter. Okay. So we talked about the two kinds of chromatin. The euchromatin that's relatively open for expression, and then heterochromatin, and there are two kinds. And the heterochromatin generally is inactive, mainly because it's so tightly condensed. You have the constitutive, these are structural areas like centromeres, telomeres, and then facultative. The best example of that would be the X chromosome in the mammalian female. One is shut off, and it's sort of a random choice. So it's not the same one in every cell of the female. It's a decision that happens where you get both expressed, but in a particular cell, there's only one active at a time. And then I updated, and actually I purchased this image of chromatin to get rid of that sort of outdated one in the notes. And here you see these two general classes in the interphase nucleus. This is where there are no condensed chromosomes, but yet you see these different density regions. You have the nucleolus, of course, which is very prominent. You can see this with a light microscope if you just look hard enough. You'll see kind of a little dot inside the nucleus of eukaryotic cells. This is where this is the nucleolus, and it's where the ribosomal cistrons reside. And then you can't see any distinctions with the light microscope, but with the electron microscope, you can see regions of very dense chromatin and other regions where it appears to be not as heavily condensed. And the kinds of particles that you see in these two general areas here are a 10 nanometer fiber and the 30. Even the 30 in the euchromatin region, the region that you expect to be more open and ready for transcription, you'll see a mixture here. In heterochromatin, generally you see a 30 nanometer folder, but what I'm going to tell you in just a few minutes is that maybe a 30 nanometer fiber is really a figment of our collective imaginations. So this is kind of the view that you have a continuum of structure going from the 10 nanometer beads on a string up to the next highest level, which is the 30 nanometer, and you needed to add histone 1 in order to go from a beads on a string to the 30 nanometer fiber. And then, of course, the 30 nanometer fiber went into successively higher folding orders to eventually make a condensed chromosome. But before we get into that fairy tale, let's talk about something else that's very interesting that has come along in the last several years that was not something that people were aware of when I was in graduate school and trying to learn all this stuff. It's the concept of chromosome territories. In the interphase nucleus are these linear DNAs which come together to make chromosomes, but when they're in the linear form, opened up into regions of heterochromatin and euchromatin, are they all tangled up, or do they have boundaries? Are there territories assigned? And it appears to be the latter. There are territories within the nucleus. In other words, the relative positioning of the chromosomes is fixed. And here you see they have fluorescent tags where they've been able to label the different chromosomes and with fluorescence and construct these three-dimensional maps. And here you see a two-dimensional view of the kind of information they were working with, but they would take optical sections through the nucleus. And quite remarkably, each of the chromosomes appears to have a position with respect to who its neighbors are. In addition, these are all, it seems to be that a lot of the action takes place close to the nuclear matrix. Transcription, gene expression is happening apparently close to the nuclear pores. That makes sense in terms of logistics. You want to get those messengers out into the cytoplasm after you've processed them and get them on ribosomes so they can be translated into protein. But anyway, this idea that particular chromosomes always have the same neighbors. This also has implications in gene expression, because can a transcription enhancer element on a promoter from one chromosome influence transcription on another chromosome? Can cis elements act in trans? There's some indication that in eukaryotes we've thrown out the rules and we have risen or developed, gone up to the next level where maybe we can do that. Plants haven't learned to do that yet. Probably never will unless somebody gets a grant or just somebody in their garage, because it would be fairly easy to introduce that concept to plants. Oh no, these are dangerous times when the monkeys have the key, the monkeys have the code. What mischief. Anyway, chromosome territories, fascinating concept. Back to the old view of stuff. The fibers, but now we've gone from interphase into how do you make a condensed chromosome? Is it just a continuum of higher folding orders? And that was the old view. And sort of at the basic level, we talked about the 10 nanometer fiber. And if you coil the 10 nanometer fiber on itself in a regular coil, it forms what was in the terms of geometry would be called a solenoid. But it looks like you can get structures in vitro that you can characterize with a repeating structural unit that has been given the name zigzag two-step. So this is the more modern way to think about that next level of folding up beyond just one step above nucleosome beads on a string. You also have different regions of the chromosome that have a structural purpose. The centromere is an attachment point for microtubules, so you can segregate the two copies each into a daughter cell. The telomeres are very special, and they have to disguise themselves so they don't look like the ends of DNA, of double-stranded DNA, which of course they are. You don't want non-homologous end joining everybody together into one big dancing circle of those little dolls you cut out with a newspaper and they all open up. You wouldn't want that. You just wouldn't. Okay, this is what a chromosome looks like. And if you zoom down, you can actually see things that have the dimensions of like that and that of 30 nanometers. And then you can see those apparently folded maybe to make something that's 50 nanometers. Is that a repeat structure? It's hard to say, but people have postulated at least that you went from the 30 nanometer to perhaps 50, then to 200 and so forth until you have a fully condensed chromosome with the two sister chromatids. Okay, so this is pretty much the old way of thinking. DNA, beads on a string. The fundamental repeating unit is the nucleosome. In eukaryotes, it's an octamer, 8 subunits, 2 copies of 4. And then that in turn forms 30 nanometer fiber in the zigzag 2-step. And then that forms something that then forms something else that then leads to fully condensed mitotic chromosome. So let's now kick the foundations out from under the building, under that nice beach house where the car's parked underneath. We're just going to go and start shaking those poles and see the family up top who've rented it for a week, whether they come running out or not. This is, of course, from the July issue of Science, and this is actually a piece of art here. An artist has a little place online. You can see the artist actually making a painting here and put metallic paint on it, and this is sort of an artistic reconstruction of what it really looks like. But this issue of Science has a couple of articles that knock on those pillars of our understanding. It looks at the structure of euchromatin and heterochromatin. It does not look at the structure of a fully condensed mitotic chromosome, though. But it does look at euchromatin, open, and how that differs from heterochromatin. And what they found was there's no difference in terms of higher folding. It's just more dense version of what appears to be random association. Just a tighter ball of string that's not wrapped in any particular way. What we think is that regions come together in euchromatin. They're attracted because they are decorated with proteins that have affinity for each other. So you could get loops potentially forming because they're outside of the histones. The histones themselves attract complexes of proteins, and so you could get regions that would naturally be compatible. They would have affinity, and they would form these little sub-organization modules. And you could increase that tendency going from euchromatin to heterochromatin, but there's not any higher folding order that's going to lead to a condensed mitotic chromosome here at all. And so let's see whether we believe it or not. Hope this link works. This is for, well, good. This is what it looks like. This is the actual supplementary information from the paper. And so what they've done is they've used a technique called Chrome EMT, which is a way to cross-link a dye directly to DNA, and then that allows you to coat it with a metal, osmium tetroxide or something, that gives it opacity in terms of the electron, so you can actually see it. And so we're looking at not anything isolated in the test tube, biochemically purified. We're looking at its in situ state. And we're able with these new techniques in electron microscopy to take optical slices like a confoculus that we're doing with an electron microscope And we can get a three-dimensional picture of the actual organization of euchromatin. This is where genes are actively being expressed. So if we run this little movie, you'll see it going down the slices. And so I'm going to show one of euchromatin, and then I'm going to show one in the same kind of cell that's heterochromatin. And you'll see there's no basic difference. You don't see any 30 nanometer fibers at all, in fact. The fibers seem to be ranging from 5 to 24 nanometers. There's not even a 30 nanometer fiber in euchromatin, according to this evidence. So let's watch this a minute. This is as you're going down into the cell. You see the nuclear pore up at the top there. Now it's going to go back again, back up through it. Okay, let's get out of that. So that is euchromatin. It's a mess, right? Let's look at heterochromatin. Unexpected error has occurred. Well, isn't that lovely? Okay, this is heterochromatin. You can see it zeroing in on heterochromatic regions. That might, I need to read the article again. Is that a condensing chromosome? Because I thought I saw some microtubules there. But what you'll see here, you don't see any higher orders of folding. It's the same, it's just more granulated. So if this is the evidence, now we have to build a hypothesis to explain it. All those people before you that went to medical school thinking there were higher order folding, do you want those guys operating on your brain? I don't know. Risky business.
So is the heterochromatin located more centrally in the nucleus?
It's actually, you see it sort of spotty through the interior, but there seems to be a distinct concentration around the edges also. So you got both heterochromatin and active euchromatin, sort of paradoxically. The euchromatin is seeking out the pore regions, but yeah, you'll find it in both locations. Okay, so that's the evidence. So you see that's a little different than probably all the textbooks. This is from the same group where they have an overall picture. They are, if you'll notice, they are trying to make the link all the way from euchromatin, heterochromatin to condensed chromosome. I don't know how tight that link is. I'll just have to do some more reading. Okay, chromatin is flexible, disordered, 5 to 24 nanometer granular chain. That's how the new way of talking about it goes. No higher order folding patterns observed. No 30 nanometer fiber. Organization patterns are the same between interphase and chromosomes. That last point I'll have to confirm for my own good. Okay, So that's the new material, and I thought it's such a significant change in the way we view things that it should definitely be included here. Other things that we'll go over these, we've been over this. The early geneticists were trying to get clues about how genes were organized in the genome, and various stains were used, and you see banding patterns. So one is to directly stain the DNA, and this G banding was seen. It usually is thought to identify GC-rich regions. Turns out most of the active genes are not in the GC-rich regions, or not in the bands, but it implied there was some kind of, that the genome was not uniform. Then we looked at lamp brush chromosomes, and we talked about them being found in amphibians, the newt, in fact, even in the eye of newt. We talked about lamp brush chromosomes, and this slide, of course, should have an exam 4 alert in red on the bottom, and about how you could possibly just look at a picture similar to this and give me the direction of transcription. It would go from the short branches to the long branches, with each branch representing a transcript. So you have lots of RNA polymerases simultaneously transcribing this thing, thusly. And the fact that there's a looping type of organization, that still holds. Here you see some of the older views here of 50 nanometer fiber. So, but this is with an intact chromosome, and I'm not sure, I mean, how do you deal with this? You just throw this observation out? So what I think is that the isolated proteins have a tendency for higher order folding. It's built into their structure. Proteins crystallize with themselves without ever having done it in nature. They do it as you prepare them for X-ray crystallography. So I think that it depends on what you decorate the histones with, whether you inhibit that higher order folding or not. In euchromatin, heterochromatin, you have lots of proteins that are trying to establish that state, and I believe they're getting in the way of this natural crystallization process that will transfer their present structure into a condensed mitotic chromosome. You just change the local protein partner environment, and it just happens automatically. But we'll see whether that has any validity or not someday. Okay, the polytene chromosome is the other general type where we learned about the organization of DNA, and we gained from that the more open, physically, if you just look at it, the more open, the more likely it is to be expressed. And you remember that's where you've gotten an internal duplication of the genome without the cell dividing up to 9 times. And it's in the diptera or the Drosophila, the fly larval stage in the salivary gland. And here you see this banding pattern. And originally they thought, well, maybe the banding pattern represents genes and intergene regions, but it's not that simple. But what we do see here are regions that are more expanded, and these are called puffs. And some of the first observations with puffs occurred in the heat shock response genes. If you take any organism, whether it's bacteria or mammals, and you suddenly raise the temperature 10 degrees centigrade, you get a drastic response to protein unfolding. And in plants and higher eukaryotes, you can shut down most gene expression, and just with a very small, relatively small subset of genes get tremendous overexpression. And we began to study that in the 80s, early 80s in my laboratory. It was called the heat shock response, but it was first observed in Drosophila with these puffs. These are expanded regions where apparently, even in the state where you have parallel copies, when you start expanding and decondensing, naturally that region's going to have to puff out. And we've identified puffed out regions with the location of the heat shock genes. Of course, there are other kinds of puffs, and now we know about lots of puffs. And here you see transcription is happening both in the band and the interband. So much for a good idea, a nice little hypothesis. Facts got in the way. This is probably P32 labeling, UTP. So you're looking at transcription. This down here, this is like puffs gone wild, right? These puffs are puffs of puffs. When I was a graduate student, oh, here goes another diversion. I've told you about the Greek skeleton of the ground sloth at the University of Georgia Library that would salute every morning as I went into the stacks. But my first question when I went to meet with my PhD advisor, I was so in awe of him. He was head of the whole division of biological sciences. I had to get an appointment to meet with him. The deal was I was going to read, and then at the end of the week, I'd like to discuss my reading with him. I just didn't want to wander helplessly through the stacks on my own. And my first question was, "Oh, great and noble and on high Dr. Key, beloved benefactor and mentor, I have a question. What is a Balbiani ring?" And here it is, and I've included in every lecture since, so that you won't be faced with that awkward question. A Balbiani ring was named by Mr. Balbiani. I don't know if he's Johnny Balbiani or what his name was, but he discovered these things, and this puff of a puff of a puff. It's a very active region of a puffed polytene chromosome. There you have it. Not much left for me to tell you about my life. Okay, where would you go looking for lamp brush chromosomes? The dumpster of a large supermarket? Under a rock in a stream in a dirty laundry hamper, perhaps? Near a deep sea thermal vent? Wherever there are amphibians, hopefully they won't be in your laundry hamper. Next topic, centromeres. This is a region that is constitutively heterochromatin. It's a structurally designated area for the attachment of microtubules, so in anaphase you can pull the two newly replicated copies of the genome apart and segregate them to the sister, to the daughter cells. Movement is by microtubule depolarization. What does that remind you of? The microtubules grow and they attach to the centromeres, and then they start depolymerizing at their far ends, and it pulls the chromosomes apart. Doesn't that sound like bacterial conjugation with the sex pili? It goes out, makes contact, then it starts to depolymerize inside the host, and it brings it in. That same process happens. So you need some kind of organizing center at the two opposite poles of the cell just before it divides. Some anchor point where the microtubules now can pull against, provide resistance as they pull the two chromosomes physically apart as they depolymerize now. These centers of organization are at 2 points, as I just alluded to. It would be the centromere is where they are attached, and the other end of the attachment is the centriole. That's where they do the depolymerization. So the term microtubule organizing center is often used in the textbooks, but it refers to these attachment regions, one being the centrioles and the other the centromere. Plants don't have centrioles. Uh-oh. How do they do it? Nobody knows. Centrioles, these things plants don't have, nor do fungi have them. But there you see them outside here at the two poles. They're the other attachment end. Here you see them represented here. There are two of these bundles that sort of look like the end point of flagella in the spirochetes, probably where they came from. Metazoans have them. I had to look up metazoan. Metazoan is an animal above SpongeBob. He didn't quite make it into the club. Back to centromeres. This is the stages of mitosis here, and anaphase, of course, is where they split. One of the words to remember is cohesin. Cohesins it's actually a complex of 2 or 3 proteins that stick the chromosomes together. So you'll find cohesins, bridges all along here. This protein is found in everything that has chromosomes. And this is what the metazoans have capitalized on. They have learned a new trick with these sticky proteins. Remember, these are the proteins in this synaptonomal complex. Plants can't do this, but animals can do something pretty marvelous, and I'm not going to tell you till we get there. And when we get there, I'm going to flash another cover of Science magazine. Make you feel bad. And make those guys in biochemistry feel bad because they didn't tell you about it. Okay. These are some of the keywords that I just installed this morning. Chromosomes are not uniformly located in the middle of a chromosome. They can be lots of places. Off center, at the telomere, or right in the middle. These 3 terms are in the key terms. Notice what happens here. Acentric fragment. If you got a break, and we talked about transposons introducing breaks, anything not directly connected to the centromere gets left behind. Well, or does it? It does in us. We'll see alternative situations in a minute. Here we have that term again, the MTO. We've introduced a new term, kinetochore. Kinetochore is a part of the centromere. Centromere is sort of a location. The kinetochore is actually the physical structure where the attachment of microtubule occurs. The kinetochore. It's a distinct structure. Yeast has one that looks a little different than the ones in other organisms. It's called the DA1. So here you see centromeres, kind of a nice generic picture. Down here you can see where it's very highly condensed. Here you see just another slide of what we've been talking about. Kinetochore, these are just pretty pictures. Notice you get this kind of a cup-like structure. At the centromere, the structure is called the kinetochore. And what we're going to see is that it has a very unusual organization of DNA in histones. And here you see the mammalian kinetochore, and you can see a very distinct kind of structure forming there. And you don't really, here you see where microtubules are beginning to attach to the kinetochore. And this is a slide that is in the key terms. This is where we kind of take this neat little textbook idea, I went to a review article for this, of the centromere being in the center of the chromosome, to the different possible ways it can be. In budding yeast, we have what's called a point centromere, one clear point. And in fact, it has a DNA sequence that we can look at and say, aha, that is the centromere. In insects, mammals, and plants, There's nothing we can look at and go, aha, that's the centromere in terms of a DNA sequence. That was a big surprise. It apparently is controlled epigenetically by modifications of the histone tails and the proteins that they in turn attract. A regional centromere, not it's kind of a region of the chromosome is designated for that, but it's not any one particular base sequence. And then look at our dear friends, the nematodes. Holocentric. That's kind of an oxymoron. Holo, everywhere is the center. So if you had an acentric chromosome, if you had, I mean, how do you have one? Because you've got microtubules attaching many, many places. Know these three different possible organization approaches. Point, regional, holo. And here we get to something pretty interesting in terms of details of organization. It turns out that in the centromere, underneath the kinetochore, the foundation of the kinetochore has a distinct pattern of histone DNA organization. You have this alternating pattern of regular nucleosomes. The octamer that we've all learned in basic biology, which we'll learn about a little more later. The octamer, you have 2 wraps of DNA around every nucleosome. Every nucleosome is the standard kind. It has 8 subunits, 2 copies of each of the 4. But notice these regions here. They have a special variant of histone 3. This particular histone 3 prohibits the formation of an octamer. It gets stuck at the tetramer. It's like half a nucleosome. It can only accommodate 1 turn of DNA around it. That's strange. How can anything work with just 1 turn of DNA around it? That's the kind of nucleosome we inherited from Mama Archaea. Only 1 turn. Archaea only has histones 3 and 4. It's not wide enough with the H2A, 2B cap on the top and H2A, 2B cap on the bottom to extend the surface that can be used to wrap. So in us, in eukaryotes, you can wrap almost 2 turns around a nucleosome because we have an extended platform. In Archaea, where it started, 3 and 4, it's a tetramer in Archaea. You have this alternating pattern of octamers and tetramers, octamers and tetramers. And that forms the foundation for a series of proteins that build on that, which in turn is a multi-layer complex, which in turn provides an anchor site for the microtubules. You need to be familiar with the histone variant. Syn H3, syn for centromere, H3 a variant, the centromere variant of histone H3. The location of this special variant, these special half nucleosomes in higher organisms seems to be epigenetically controlled. It turns out that nature was experimented a while on how to do this. And you have a couple of different strategies that were employed. But here you see Drosophila versus yeast. In each case, it's a tetramer, but the composition of the tetramer instead of an octamer is a little different. Here it's H2A, H2B, 3 and 4, but the 3 is this special H3 variant, which prevents it from forming a full octamer. This is half nucleosome. Doesn't have a second copy. It's called a hemisome, a tetrazome. This is what yeast does. It only uses 3 and 4. Doesn't have the H2A, H2B. In each case, the result is you only have room enough to wrap 1 turn of DNA around it. I will not ask you the details of this. I'm not going to ask you what's in a hemosome versus what's in a tetrasome. This is just to provide some context for you that sometimes there are several ways that evolution has gone. So let's look and dig down into the details of the centromere we know most about, and that's the one in yeast, where we actually have a sequence, and a lot of NIH money and NSF money was spent on the model. Yeast as a model system, we can learn what it looks like in yeast, and now we know what it looks like in all higher eukaryotes. In this particular instance, that turned out not to be true. But let's see what we've learned about the centromere from yeast. CEN is the name of the DNA region that corresponds to the centromere. There's a particular sequence. It's about 120 base pairs in length, and it has 3 domains, CDE, that's centromere domain, element 1, 2, and 3. And you need to know which of these is the most critical, which is the most highly conserved. If you put a one base mutation in one of these 3, you get improper segregation of the chromosomes. And that turns out to be 3. That's 11 base pairs. You mutate one base, you can mess up segregation of the chromosomes in yeast. What comprises, we go back again just to try to see whether I still have a pulse. What comprises the MTOC? Let's see. Centrioles and centromeres. Yep. Number 1. Okay, back to the matter at hand. Here you see the organization, element 1, 2, and 3. Element 2 is the largest, but it's not very specific. It's just 90% A's and T's. But these are specific sites here, 9 base pairs and 11 base pairs. These are really bookends on something that's just generally AT rich. These 2 places are where specific binding events occur. You get centromere binding factor 1 at element 1 and centromere binding factor 3 at element 3. In the middle, this is where you get that histone 3 variant. And in yeast, it's not called SYNH3. It's got its own little yeast name, CSE4P. 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 It's like the conga lines. You wind your way through Lillian's. Oh, that's where old people go. Let's see, where's that other place? Oh, the Dairy Queen. Yeah, you do a conga line through the Dairy Queen parking lot. Trying to ease up to the counter to order that Georgia mud fudge or whatever that is. Boy, that is good stuff, isn't it? Man, I'm glad I didn't know about those as a youth. Okay, I'll digress again. I got to keep moving. Intrinsic binding. AT-rich, remember the up elements, AT-rich, it has sort of a curvature to it. This has some curvature. It turns out that curvature probably helps in this case because that central region wraps around one turn around this histone variant, but it's really wrapping around that full tetramer of the nucleosome. In this simplified figure, it looks like it's just wrapping around H3, but it's really that tetramer nucleosome that's wrapping around. And you get the 2 complexes at either end now all come together to form this gigantic complex of CBF1 and CBF3. This now becomes the foundation, the bedstone that you start building your kinetochore on. The human analog or homolog to CSE4P is SYNPA. Those are key terms in Module 4, SYNPA and CSE4P. Here you see the centromeres, the points of contact, the microtubules, the centrioles, the microtubule organizing centers, And this is an anaphase where they're beginning to split open. And this, I just loaded that this morning. It looks like a brain after eating some barbecue wings or something, doesn't it? Like, boy, that was hot. This is a bouquet, or as they say in Bronson, a bouquet. A telomere bouquet. Those people in Bronson, they all are Second Amendment people. I should not speak with such irreverence. Okay, so here you see in prophase, going into meiosis, gamete formation, reduction, division, all that stuff, prophase, you get all the ends of the chromosomes come together to make a nice little bouquet called the telomere bouquet. And the ends of the telomeres are actually attached to the nuclear envelope. And in some organisms, the nuclear envelope begins to degrade and they connect to the centriole that's on the outside of the nucleus. It's like a magnet. They seem to be attracted to the MTO, the centriole, or organizing center is what you'd call it in a plant. And they are attracted like, iron filings on a piece of paper with a magnet on the backside. The telomeres come together, and here you see them. They are fluorescently tagged with proteins specific to the telomeres. You can see in these phases here, D and E especially, they're all drawn together. This observation was made by just under the optical microscope in, I think it's 1828. It was called a bouquet at that time. And they didn't know what it did now, and we still don't. We know some more about it. It doesn't go away unless you've undergone recombination. It seems inherently to be involved in aligning the paired chromosomes. After the division, the sister chromatids, that seems to be reasonable, but there's mutations. It's hard to get that nailed down. And you'd expect maybe the chromosomes to start zippering up from the telomeres because they are joined together in close spatial arrangement. So that bouquet is still a puzzle, but it happens, and it's a nice concept. Which protein is found at the SIN? Which one of those is actually a histone variant? It would be, I'm hoping number 2. Whew. That's the end of this little portion here, but not the end of the fun we're going to have today. I had to say that fast because I could hear notebooks slamming all over. What's the time? 1:43. 1:43? Do you want to take a little break right now? Just a 2-minute break? And we'll start on telomeres. I know you're going to come running back for that. Okay, so why are telomeres special? As we've said before, you have a linear piece of DNA for one thing, and it has special problems in being replicated. You always leave off some. It's hard to start replication right at the very end nucleotide of the template. We talked about how adenovirus gets around that problem. But telomeres have a special way to get around that too. They know how to keep their telomeres young and fresh, but they don't. I don't know whose side they're on, but they only do that for the gametes. The rest of the cells, they go, yeah. It's all about the gametes. And the gametes are going, it's all about the DNA. It's not about us. It's like billions of us or something. I don't know. Anyway, telomeres. They're at the end, confer stability, meaning that they masquerade the end so it doesn't look like an end, so non-homologous end joining won't use it as a substrate and connect all your chromosomes together into one big molecule. They shorten every time the cell divides, 50 to 100 base pairs per division. That is so sad. It's one of the things that can kill you. Oops, running a little short on telomere over here on chromosome 26. Consists of a long series of repeats. It's a very short repeat. Notice 100 to 1,000 of these repeats. And it's repetitive, it doesn't code for a thing. It's throwaway. It's throwaway DNA. It's so that they can get shorter and shorter and you're still not deleting or damaging any genes. It's sort of buffer DNA on the end. So you can, in somatic cells, you don't need to worry about the fact they're getting shorter every time they divide because you don't care. You drive real fast, go around curves, hit trees, it doesn't matter. Just get those gametes out. What is the sequence? Well, it turns out that one strand, there's an unequal distribution of the G's, the GA, and the GTs. Oh, I got the GTs. The GTs versus the C's and A's. We'll see how that works out to the advantage. There's a G plus T rich strand and there's a C plus A rich strand. The G plus T strand is renewed in the gametes. Most somatic tissues have no enzyme called telomerase to keep growing repairing the telomeres to replace those repeated sequences that got left behind in the last DNA replication. Oh yeah, I didn't tell you, there's an enzyme, telomerase, that actually its sole function is to grow those ends right back out again. So there's no damage, so your gametes will live forever. 90% of tumors have that enzyme because tumors, HeLa cells, there's some changes that happen when a cell line is immortalized. And this is one of them. The telomerase enzyme is expressed all the time now. That's one of the things that you need to do if you're going to make cells immortal. Some people in the back taking notes. Every year there's somebody who wants to do an undergraduate science project. I don't know, they think it's going to take a long time to get their degree or something. They want to immortalize human cells. 90% of tumors have this. A lot of cell lines, only last a certain number of generations and they kind of die out. That's because one of the reasons is the telomeres are getting shorter. Telomerase inhibitors show promise as anti-cancer agents. Because of the fact that that's one of the things that often gets expressed in a tumor line. What do you need to do to immortalize a cell? Well, one of them is to introduce the telomerase gene. If you were in the lab doing this as your undergraduate project, your major professor thinks you're working on strawberries, but you're doing something else. One of them is you just ordered the HTERT gene. Human telomerase something repeat, who knows what it stands for, but HTERT is a key word on module 4. It is the human telomerase gene. It also requires an SV40 T antigen, something to help it along, but if for exam purposes, if I ask you how to immortalize a tissue culture line, I'm really looking for express the HTERT gene. Normal tissues can be transformed to make them immortal by HTERT and SV40 antigen and another protein. These look like viral derived proteins, certainly SV40 is. But HTERT is the one that you'll need to know for exam purposes. So let's get the details of how this works. Here you can see the repeat structure of the telomere itself. Notice you have mainly C's and A's in one strand and G's and T's in the other. There's a few proteins that you'll need to know, and I've put these in bold. What you're going to need to do is, if you have this, if you want to grow your, if you want to protect the ends of your chromosomes, where the telomeres are, one thing you want to do is hide those ends. And what the cell does is sort of use a mechanism of recombination, where you have strand invasion. The 3' end of your chromosome is made single-stranded, so it looks like it's going to be involved in homologous recombination, and then it doubles back on itself and displaces. It actually invades itself in the region of the repeat. So it really is homologous recombination happening at the ends to form these telomere loops at the end of your genome. So you need to have a protruding 3' end, and one way to do that is to chew back the 5' end. And the protein that keeps that process from getting out of hand is STN1. There's another protein involved here, CDC13, and it binds to STN1. It recruits that, and it recruits telomerase. So it's kind of the organizing protein. CDC13 recruits STN1, which is part of the processing to make sure this chew back of the 5' end doesn't get out of hand, and then it recruits the enzyme that's going to grow one of those two strands. It's important that you know which strand is grown. Of course, it's the strand that corresponds to the 3' end of the DNA, and that will always be the G plus T rich strand.
So if the ends of the telomeres are repeated consensus sequences, how does the CDC13 know where to bind? Like how far down on the...
Okay, next question. Anybody have another question? I have no idea. You're not allowed to even think that question. I don't know, we don't want to talk about it. This never happened. I can edit this, right? I hate good questions. It's the enemy of all learning. Okay. Just accept it. Just walk along. Yes. Yes, Professor. Okay. Now the enzyme. This is going to be some crazy stuff, but not that crazy. The enzyme is telomerase, but it has attached to it, and you can see right there, that's part of it. It has attached to it a piece of RNA, which I think is about 192 bases long, 192, 196, but it's more than just this. But one little region is a copy of the repeat. It's RNA. That, notice, is a copy of the other strand, right? That's a copy of the C plus A strand. That's the bottom strand in that diagram. That's the strand that does not grow. It turns out that piece of RNA is covalently attached to the protein, the enzyme telomerase. Why would that be? Well, you're going to grow the end, grow it out in air. You're going to grow it where there's no template. Telomerase brings its own template and attaches it to the end. It's an RNA template, and it lays down DNA. What kind of enzyme does this make this? Reverse transcriptase, a eukaryote reverse transcriptase. Telomerase. Covalently attached is 192 bases, okay. The template RNA contains within it two of these repeats, but it's the CA-rich strand. Let's see how this works. First of all, here's the problem that arises. On the lagging strand, you have to continuously reinitiate the lagging strand, so you end up with a region here that never gets replicated, and that's why it gets shorter and shorter. So here's the way you solve that with a telomerase. This is the repeat. The 3' end here, that is the T plus G rich strand. And this is telomerase. Notice half of the repeat is able to pair at the end with itself. And now this region extends, and that now serves as the template, so the 3' end can grow all the way out to the end to form this. It adds the G's and the T's and then ends with a G. And then this enzyme comes off and binds again, right in the last 3 bases, and just leapfrogs, building one repeat at a time. So here you see it's just, this is where it was paired, and this is the new part it just added. Now it translocates and lines up again to do the same thing. Pretty cool. So now you've got the 3' end extended way out here. Eventually you're going to have to fill in some of this. How would you think that you could prime it going back the other way? This is probably what you're not aware of, but you will walk out of here in awe. G can pair with G. Cool. That's why this is the G strand, the one that grows, the one that you want to be single-stranded because of the unusual base pairing properties of the G. Those unusual properties cause it to form a G hairpin, so it can be primed. And also they cause it to be able to form a very weird type of structure that disguises the end, so it doesn't look like an end. It's sort of a cloverleaf pattern of, it's hard to describe. We'll see that in a minute here. The G quartet. I should have that in bold. So you can get this G hairpin forming, which is where G goes on itself, and this is useful for priming, but it also forms this crazy structure, this kind of a clover leaf looping out structure with itself, again, to disguise it like there's no end here, no end here, this doesn't even look like DNA. Non-homologous end joining goes, it should move on, there's no end there. This is how that G quartet forms. This is the repeat. The 2nd, 8th, 14th, and 20th G loop in together by hydrogen bonding to form this weird looped out structure called the G quartet. It's quartet because it stacks sort of in repeats of 4. Okay. So that's pretty cool. You probably never heard of that before. How does the telomerase move? Does it have tiny little feet? Sits back down? How does it move? What kind of mechanism do you think there might be at the molecular level? Is it random motion, like MUT-S just kind of slides one way, then kind of goes with the tides, like seaweed? Or is it have a polar movement mechanism? It turns out it does. It's always headed towards the end. And the way we think that operates is there's a protein that when it sees a G quartet or a G hairpin, it has an affinity for it, and it just sits down and goes, oh, for me, yeah, G quartet, love this. Sits right down, and when it sits down, it pushes the telomerase towards the end. The telomerase just made it, you know. It's a G thing, and then, oh no, this protein comes and goes, perfect place to sit down. The protein's the HNRNPA1 protein. It makes a perfect landing spot for when it sits down there, and when it sits down, it goes, ugh, okay, and then it makes another one, and then it sits down again. It just pushes itself by just the affinity of this HNRNPA1 protein. It's thought to just physically push the telomerase one more repeat distance. Here's the what's it called? Has a name. It's the T loop, I think. It says D loop, but it's a displacement loop. T loop, there it is up there. The T loop right there is this loop I was telling you about, where you get sort of a recombination event occurring where this 3' end here invades itself and displaces and actually forms a D loop. And so you get the end of the chromosome before, you know, after it's formed all of these G-type structures. Eventually that goes away and it actually gets hidden by sort of recombining a Rec A type of movement, except it doesn't use Rec A to strand invade. It uses TRF2. That sets up the condition so that this really very specialized kind of strand invasion can masquerade the end of the chromosome, the T loop, TRF2. What good are telomeres? What happens if as you delete them? Well, I'm going to show you some examples here from Arabidopsis, a plant. It says if you mutate the telomerase gene in Arabidopsis, it can survive about 10 generations, and then things begin to go downhill. And you can see here the generations, and this is a gel with hybridization probes that are specific to that telomere repeat, and you see they're getting shorter and shorter and shorter. They shorten about 500 base pairs per generation. In plants, before we get the last couple of slides here, got one of these stupid questions. Which two proteins bind the CDC13? I think that's going to be STN1 and telomerase, if I remember correct. Oh man, looks like I missed it. Maybe I did, maybe I didn't. I have to go edit that slide because I took out some stuff and I can't tell you about it. So we keep moving. I'll check on that slide again. You don't need to know this. This is just showing you the bad effects of the telomere shortening per generation. Notice that in mice, mammals can only survive about 6 generations before you start getting bad things to happen. Here you see some plants at the various generations after putting in the mutation. Generation 8, not too bad. Generation, you know, it gets worse and worse. 8, you're beginning to get some effect. You don't need to know the specific numbers or anything. Here's some more of that. It has all sorts of developmental effects. It's not just that the plants are getting shorter, but you're beginning to eat into genes, to things that you need. They don't grow back except in the gametes. Okay. So the telomeres, if we kind of jump back out at a greater distance and look at it, these are a special place where heterochromatin is always present. This is not to be expressed except with these little repair operations. But sometimes when you have a gene that is transposed to some other location or is just flipped around from one orientation to another, you'll get too close to the telomere. And if the telomere starts, if the heterochromatin that naturally covers the telomere begins to creep, you can get the shutdown of adjacent genes. And this is a famous example of it in the Drosophila eye color. The eye is naturally red, and the gene that was mutated is this red eye gene. But of course, the gene is called because of the phenotype they saw when they got a mutant. They said, look, the eyes went white. It's the white gene. No, that's a mutated red gene. It's that pigment gene, and you'll notice that where you're beginning to get the heterochromatin creep in the, depending on whether the gene is oriented one way or flipped around the other way, where the promoter is close to the growing, creeping heterochromatin. So to get back to that, the idea of heterochromatin and constitutive heterochromatin, how it may get organized and put in place. And these are some of the big questions of regulation. You know, what controls the transition from permanently shut down heterochromatin to always open euchromatin? These are big switches at the regulation level that are very important, and you have to be careful how they're regulated. So we know a little bit about these processes studying this kind of effect here in Drosophila, where a gene got flipped a little too close to the telomere. So we know some of the stages in forming heterochromatin at the telomere, and that's seen in the panel on the right. There's a protein called RAP1 that will end up on a key term list. It binds to the actual repeat itself, RAP1. That, in turn, has affinity for a couple of proteins, the SIRs, SIR3 and SIR4. They go together, and these are sort of some of the foundation proteins of heterochromatin. And they begin the nucleation by RAP1 actually binding the DNA directly and saying, aha, this is where we should begin. This really is a telomere repeat. Because you don't want RAP1 binding to some crazy place, because you could shut down whole banks and loops if this got out of control. So you have to have something very specific for it to recognize. So it recognizes repeat, and it triggers the binding of the next series of proteins, which is SIR3, SIR4. They have affinity for themselves, and they begin to bind to the tails of the most conserved histones, the 3 and the 4. And they have affinity for themselves, and it's kind of a zippering up cooperative effect. They begin to coat the tails of 3 and 4, and then it turns out that 3 has an internal domain that mimics one of the major matrix proteins of the nuclear matrix. And this is why heterochromatin is often stuck up against the matrix on the inside of the nucleus, because SIR3 has a little internal domain that looks like lamin. Lamin is one of the proteins of the matrix, so it just kind of sneaks in there with all the other lamins, and everybody's going, cool, we're all lamins, right? Yeah, we're lamins. It just nestles right in, dissolves in the matrix for that reason. So you see this twin attachment to the nuclear matrix and to the nucleosome, bridging the two together, the SIR3, SIR4. See what this does. RAP1 recruits SIR3. They interact with histones, polymerize. Of course, this begins to shut down gene expression. And down here, SIR3 has an internal domain similar to a nuclear lamin protein. That's what helps tether it, heterochromatin, to the nuclear matrix. And well, look what we've done. We've actually completed an instructional unit. That's an odd feeling. How much time do we have?
3:13.
3:13? We have lots of time, right? 4 minutes? 40 minutes. Then people going, really? It's Thursday, people. This is the day where you need a superhuman bladder, yes.
What prevents G quartets from forming in other places in the genome other than the telomeres?
Well, you have to have a single strand. So not very often do you have DNA single strand except when you're in the act of replication, and then you get immediately coated with single strand binding protein and things like that are happening. So if you have a special environment at the telomere to exclude single strand binding protein, then you can get these novel things happening because you can maintain it in a single strand state long enough to form the G quartet and the G hairpins. You won't be allowed to come to class anymore. You should just take a distance. Just kidding. Okay, let me go to the log. So I didn't know I would get this far. But I definitely need to keep going. How do I get to? How do I? Oh, down here it must be. Where's a? Oh, here it is. So the histone chapter is not very long. I don't go through the whole chapter. I just want you to know the basic structure. Somebody told me that if you made your password 18 characters long, you'd never have to change it again. That's not right. And now I've got this tremendous 8. So it take me a minute here. Let me see, how does it start? No, don't really. Oh yeah, I still got it. But I'll be changing it in about 2 more months. You can never get a password shorter than the last one you had, of course. Look, there's no histone one there, is it? Surely there's a histone lecture. No, because I went in yesterday and took out the histone thing. I'm going to have to go here and get it back. That's what I get for trying to keep the website updated. Well, that's nothing but the exam. I wouldn't want to show that. Might as well, what's in there? How do I open this up so I can see the full titles? Let's see here. Well, I don't see that I've got histones, so let's move to the next topic. I think maybe I should go back and put some, but we'll keep moving. Eukaryotic transcription. If I could only read the last part to see what the title was. HD mediator regulation. Where are parts 1 and 2? Let me see. Oh, it's probably at the website. Of course, So I'll have to... Okay. In the beginning here. What? You kidding me? Who's running this outfit? Okay, this is working out. This is working out here. Where's Summer? There's Summer. The boys of Summer. Module 4. Yeah, this is before the course designers took away my creative license. What got histones and nucleosomes? Look at that. Chromatin, let's see what that is. I bet that's the one. Oh man, Dr. Gurley, when you... Well, if this is the histone one, it'll get moved there. And next week we'll get into... Yeah, this is the one I didn't get a chance to move there because I thought I wasn't going to get that far this week. Okay. What we're going to talk about now is the nucleosome structure. And I'm going to try to make it so that it's just the sort of the meat of it here. Of course, we have the euchromatin, which we've talked about. Half the protein mass of chromatin is protein, and half of that, let me see, let me go back, I'm just muttering here. 60% protein, 30% DNA, 10% RNA. Half of the protein of that 60% is histone. This is the molecular structure of the nucleosome. This is the best understood, and this doesn't seem to be going away with the sands and currents of time. This is a nucleosome. This is an X-ray crystal structure derived, so you'll see as they crystallize together, you often get pairs like this. But what you can see is that DNA is wrapped around the outside of a cluster of proteins. If you look down on the top, you'll see this. It doesn't mean much at the moment, but actually if you look down on the top, you see poking out here between the strands, you see poking out unstructured tails that are waving around, and these are at the N-terminus, and that's where all the epigenetics happens. The nucleosome. If you took just about any eukaryote and just tickled it with DNase, just enough so on average there was one cut per chromatin DNA strand that you had, and then you cut it with a restriction enzyme at a particular place, you would end up with a ladder of different sizes, and the steps in the ladder would be every 200 bases. And so we know from that the spacing of nucleosomes is about every 200 bases. It's not actually wrapped around 2 times. You can see DNA is wrapped around 1.67, but people like to say 2. 3 and 4 are the most highly conserved, and they go all the way back to archaea. Non-histone proteins, HMGs, you don't need to know any of this. 10 nanometer fiber. You need to know this. We're just going to hit the high points of this because this, not all of this will be on the exam. The packing ratio, here we are again, is about 6. What does that mean? If you have DNA stretched out linear with no histones on it and wrap it around one nucleosome, the size now will be 1/6 the length that it was originally. So that's the packing ratio, that ratio between unpackaged and packaged. So that's the first condensation step. 30 nanometer fiber, we now know that that can form in vitro. It's certainly real, but does it actually form in euchromatin with all those other proteins all around it? We know that there's evidence now that maybe it doesn't. It may form in the formation of the chromosome. We're not sure until we do some more reading on that. But anyway, in order to go from the nucleosome to whatever that next level is, requires adding another histone. You know, there are 4 histones. This is the 5th histone. But paradoxically, it's called histone 1. It's one of the least conserved of the histones. You have 3 and 4 being the most conserved, going all the way back to archaea. And archaea, and they form together in the assembly of a nucleosome. They go together and form a dimer. And one wrap of DNA can go around that in archaea. There are 2 other pairs, and they're histone 2, but 2 forms of 2. 2A and 2B. And 2A go on the top, and 2A, 2B go on the top, and 2A, 2B go on the bottom, and you'll see that in just a minute. But this H1, if you don't have H1, you can't transition from the 10 nanometer to the 30 nanometer fiber. If you get to the 30 nanometer fiber, you have a packing ratio of 1,000. That's a big increase in condensation. Here you see the beads on the string. Always associate that with the base level, the 10 nanometer fiber. Archaea even has something that looks like beads on a string, and here you see the archaea representation. You'll need to know the individual histones, but not what they look like, except when I show you here. So there are 2 copies of H2A, 2 copies of H2B, 2 of 3, 2 of 4. They make this cylindrical thing that forms the core nucleosome, and H1 is added in between nucleosomes. It's in that linker region, and it has a regular helix turn helix domain, as if it was a transcription factor or something. Let's see, there's going to be 200 is the length, the spacing between nucleosomes on average. There's another number you need to know right here. If you took and digested overnight, you had something to do, your girlfriend wanted to go out and eat, and you got the experiment going, girlfriend, nucleosomes. You unwisely choose girlfriend. And you come in the next day and you run the gel. You'll find that the bands are not 200 anymore. But they reached a limit digestion where they don't digest anymore, and that's 146. And that accounts for 1.67 turns. So that's the core, the limit digest. If you just hit it once between every nucleosome, you'll get a repeat pattern of 200. But that includes all that linker DNA. Half the linker goes with one nucleosome, half with the other. So that's the distinction. So on the exam, maybe on the homework, I say just a very mild digestion. What would the ladder look like? That would be 200 bases on average. How about limit digestion, where you digest until you can't digest anymore? That's 146. Micrococcal nuclease is what people use. Limit digestion. And this illustrates that point. What would the average size of fragments of chromatin be if you added just enough nuclease to hit once per nucleosome? That's where you're looking for the 200. It's wrapped 1.67 times around the octamer. 3 and 4 are the most conserved. 2A, 2B show some species-specific variations. H1 can be quite variable, tissue and species-specific. H1 binds to the linker DNA. Here you're beginning to see these now kind of archaic demonstrations of nucleosome structure, but you need to know just a little bit about this. This would be the core nucleosome here, and that is H1, which binds to that little piece of DNA exposed in the linker region. So H1, so you don't have as much H1 as you have with the other histones. In archaea, there's no H1. So this is what I like. This is sort of the context that this provides. In archaea, there's no higher order than beads on a string. You never condense into a chromosome. In fact, there doesn't appear to be anything like heterochromatin. It's just beads on a string. Everything is open. Not as much as it's open maybe in a bacteria, but we don't know much about the bacterial openness. It does have a degree of condensation, but we don't know much about it. But anyway, beads of a string is the default ground state. When you have beads on a string, it's ready to be transcribed. The RNA polymerase can negotiate histones if they're in this open beads on a string configuration. So let's go to the next slide here. You're going to need to know, be able to recognize generally where things are on a picture of a nucleosome. So if you look down on the top, you're going to see a copy of H2A, H2B. If you took this thing and flipped it over and looked underneath, you would see blue. You'd see H2B, H2A, but they would be flipped around. The H2B would be over here, and then H2A would be in the center. It's a symmetrical arrangement. The H3, 3 and 4 go together. 3 and 4 go together, and then at their ends, they perform a little dance. The two H3,4 dimers join hands to form the magnificent octamer. This is looking down. There we're getting into exam figures. Let's pay special attention. See, it was worth coming back for this. This is a side view. If in the middle you have H3, H4, and then if I looked around on the back, I'd see also white H3, H4. And then on the top, I've got a cap of 2A, 2B, and on the bottom, see how it's symmetrically on 2A, 2B. It's actually 2B, 2A. I'm not asking you where 2A, 2B, but know the caps, what I call the caps. This right here, this central region, would be the archeal nucleosome. You can only wrap one turn of DNA around, but notice how it was expanded by putting an extra shelf on the top and the bottom. So now you can wrap around 1.67 times by adding a cap of 2A, 2B on the top, and on the bottom, you've expanded the surface of that cylinder. You can now compact and condense your genome much tighter. The lines, of course, represent DNA. And so here you see 2A, 2B. To me, this is not a good representation. This is 2A, 2B. This is one copy of each. 2A, 2B form a pair with each other. 3 and 4 form a pair with each other. You duplicate this, except flip it backwards, and you have the full thing. And notice to me how H3 and H3 come together and join hands like two praying mantises in a formal minuet. That's how I can identify H3. I can hear the harpsichord now at the Versailles, in the grand ballroom, and everything is glittering with gold. And the gardens are lovely this time of year. The nucleosome, the movie. Okay, let's back to really good stuff here. That is a histone fold. What? A fold is what a structural biologist, somebody who looks at protein structure, calls a series of hairpins and helices that have that form a motif, a conserved configuration or functional configuration. That arrangement of helices a helix, a long helix, and another helix. That together is called a fold. That is the entire H3 or H4, I don't know which one that is, but the whole protein forms the histone fold. We'll study some transcription factors a little later that will contain buried within them a histone fold, which is this thing. This was invented by Mama Archaea, of course. And so that would, you see here, this is the H3 and H4, how they go together. That long helix, that they both have a long helix in the middle, flanked by two little short helices. Notice how they fit together just perfectly to form the dimer. This H3-H4 configuration is mimicked in a multi-subunit transcription factor used for RNA polymerase II we'll learn about later. It looks like RNA polymerase II has mimicked the structure of a nucleosome to designate where a promoter is. It's as if evolution has said, you know, nucleosomes, they all look alike. What if I started playing with nucleosome structure and made one that had some sense, that could identify where a promoter was? And that apparently is what has happened. That transcription factor is called TF2D. Contains Tau binding protein. More about that later. This is probably an exam question. I don't know, just saying. If it was an exam question, what could I possibly ask? Well, this is obviously blue and gold. That's what everybody's DNA looks like in Gator Nation. This is the DNA wrapped around the outside. And inside, well, look, there's our friend. The minuet music is starting up again. The two praying mantises are getting to know each other. Ah, my dear, shall you lead this time? And a little too much brandy. And then you have the 3 and 4 dimer, the 3 and 4 dimer. But what are these things? Is this like a nucleosome bomb? Is that the fuse? Is that where you stick the match? These are the tails, unstructured. This is just an artist's rendition of what that looks like. It's just probably flopping around out there. This has places that can be modified by phosphorylation, methylation, acetylation. It's thought to, the pattern of modification on that N-terminus is supposed to form a code, the histone code. It's supposed to attract specific complexes for expression or repression or one tissue versus another tissue. The histone code is embedded here in the N-terminus, the unstructured. This is outside of the histone fold. This is an unstructured region that comes off the end of every one of the histones. Every one of them has a loose string coming out the end. And that is a eukaryotic invention. We did not get that from Mama Archaea. We use that to signal whether we should be in an active state or inactive state, whether we should be condensed or wide open, heterochromatin versus euchromatin. Why would you have this in Archaea? Nope. Archaea doesn't go from condensed to open. It's always open. It doesn't need any histone code. Eukaryotes invented that little string that it adds on the end of its histones and waves around out there with a signpost, like hurricane flags. We've done that as eukaryotes on another transcription protein, and that's RNA polymerase II. The large subunit, the one that corresponds to beta prime if we were in bacteria, but it has direct lineage back to the eubacterial beta prime. On the C-terminus, we have a great long tail that has no structure that just drags along behind the polymerase. that is just a signpost for the attracting different complexes that are needed specifically in processing the transcripts of Pol II. It's called the C-terminal domain of Pol II. So this is a theme that eukaryotes have sort of enumerated, this just building an unstructured domain and just wave it around out there and use it as a kind of a signpost or a binding site for specific complexes. Histone code would be there. I think this figure might be on the exam. Just saying. And this would be sort of you'd go, what is this? I thought it was going to be on the exam. That's looking at it sideways. I wouldn't do that to you. That'd be horrible. Okay, which proteins cap the two flat ends of the core nucleosome? What's on the top, what's on the bottom? H2A, H2B. The hierarchy, I could take this and just throw this in the river, right? I don't believe any of this stuff anymore. You think so? Really? No, you can't be kidding me. Oh, look at that, he's crazy. Histones have tails. How much time do we have? 10 minutes? I'm running out of material here. So there's our friends H3, the praying manti, engaged in the minuet. And here you see the strings. The histone code is this unique pattern of modification of these unstructured tails that are on each of the histones. And here you see the tails coming out in this space-filling model of a nucleosome. It happens to be H3 coming out. And here you're beginning to see some of the specific kinds of modifications. I'm not going to ask you to know all the specific kinds or even learn the code. I mean, that'd be crazy. Then you could start doing epigenetics. You could turn your spleen into a brain, and then you'd have an advantage on everybody sitting beside you. Archaea have no tails. Why? Dead men tell no tales. They just have beads on a string. They don't have to modify their chromatin condensation state. The most common types of modifications, these are the ones we've mentioned, acetylation, methylation, and phosphorylation. And you're going to need to know you need to know this list, but also what kinds of enzymes either put these on or take them off. One of the central ones is acetylation is highly correlated with activating genes. The histones near the promoter will become acetylated, and the enzyme that does that is called a HAT. You have the HATs and the HDACs. If you could just talk HAT and HDAC, you could probably get into any graduate genetics seminar. They go pass, going in. You just go HDAC, they go, you're good. You don't even have to show them the stamp. HATs are histone acetyltransferases. They put the acetyl groups on, and that's associated with activation. HDACs, histone deacetylases. They take them off. Methylases, that's the easy one. Phosphorylation are done by kinases. HAT HDACs. HDAC generally associated with repression, HAT generally with activation. In the beginning, it was thought that, well, we really didn't know how the histone code worked. At its most basic level, they figured, well, maybe something to do with electrostatic attraction and repulsion. That histones, of course, are very basic. Why are histones basic? Because DNA is negative. Histones bind DNA. But These tails are also kind of basic, and some of these modifications would reduce the basicness of them. And for example, the acetylation of lysine would do that. The positive charge is neutralized upon acetylation. Methylation does not change that. So it was figured that maybe it has something to do with how tightly you can compact nucleosomes simply by charge repulsion, doing some neutralization there by acetylation. That theory still has some merit, but there seem to be many other things that could explain it also. Phosphorylation would do the opposite. The code. Oh, tell me we don't have to know this. What I'm going to do here, so I'll need over the weekend, I'm going to pick out a couple of examples that you can go to and not have to know the whole thing. But I want you to see trees and the forest both. So I'll give you, this modification is associated with activation and this particular one with repression. And then you'll know that there are lots of others. I'm glad he didn't ask us to know all those also. I haven't picked those out quite yet, so I can't show them to you. But most of what we know involves the pattern of modification on the most conserved histones, the 3 and the 4. And you'll see that methylation can both activate and condense. So it's not, it does seem to be a code. Acetylation of K9 is specifically associated with activation. There's some of these others with activation, but you can't always predict just by whether it's acetylation or methylation, whether it's going to be repression or activation. And I will identify, also this is one that I'm not going to ask you to know specifically, just to know that there are some well-characterized ones on 3 and 4, and we'll learn a little bit more about some of these, but I'll identify these on Tuesday. This is an especially interesting place here. Lysines are often sites of modification. They can be both methylated and acetylated. But this particular lysine 9 is sitting right beside a serine. And that seems to form a switch called a methylphospho switch. It's the serine, if it's phosphorylated, can negate the signal derived from the acetylation state of the lysine. It's like this is the on-off switch that determines whether the signal imparted here on the lysine can be read or not. It's wherever you have the chance of phosphorylation right beside a place that's acetylated. You have the potential for this binary switch. We're just beginning to learn about the code. Here's another example that's not as well known. This is a lysine, and you have a threonine. Threonines can be phosphorylated also. And there's another example. I think it's lysine 27 has that. But these are some sites that are of special interest to people today, in the modern day. This is a slide from the early days. When I look at stuff, I look sometimes through different eyes. When I first saw this, my first thought was, and this is, you see the tails here, they're not acetylated and they're down, and then when they're acetylated, they're up, and it's supposed to be the chromatins decondensed. To me, that was let my chickens go, or
Chickens in bondage.
like Chicken Moses or something. It's like chickens in bondage. That was what it was, chickens in bondage. This was the chickens in bondage slide. That I saw these as little fryers ready for the broiler. And they were all wrapped up, like, help me, help me. Little arms up, chickens in bondage. I had a troubled youth. So, I don't know. I just thought I used to be weird. Okay, how much time we got here? I'm running out of my 1 minute. Let's call this off. Lights on, everybody. Yeah. Okay.